Good morning students. For standard 12 we are going to continue the chapter on mutation, and we have already started telling on that. So what we are going to discuss is about the gene mutation today. Now gene mutation is basically of two types. One is called a Frameshift mutation, another is called substitution. I told you about the point mutation already, that when if it takes it at a particular point, generally we call it a copy error mutation. One particular base pair is copied wrongly or there is something inserted wrongly, then we call it a point mutation. Now in Frameshift mutation is basically changing the whole frame or the sequence of the amino acids in the DNA. Means actually the DNA is copied in the mRNA. So that if the change occurs in the DNA, then the change automatically occurs in the RNA, and a wrong amino acid sequence will be formed. We'll come to that. When we see the examples, you will come to understand. Like in Frameshift mutation, we have two types. One is called deletion, another is called insertion. Now let us take an example of the sequence of the mRNA. This is the mRNA. Actually, it occurs in the DNA. But DNA is copied in the RNA in the process of transcription. Then through translation, the sequence of amino acids are added up to form a particular protein. And that protein is actually the enzyme which catalyzes the reaction. Now, see this example. This is a sequence of the mRNA. And the sequence of the different nitrogenous bases are like this: AAG, AGU, CCAU. Means these are adenine, adenine, guanine, adenine, guanine, uracil, cytosine, cytosine, adenine, uracil. This is the sequence. And this sequence, all these are in form of triplet codes, which you are going to learn later on. Means each amino acid is basically, it's made up of three codes of the base pair. Like AAG, this codes for lysine. AGU codes for serine, another amino acid. CCA is for proline, and another may come in the next sequence. So the sequence of the amino acid, suppose in a particular segment of the mRNA is lysine, serine, proline, like this. Now suppose there is a deletion. Deletion is that this particular nitrogenous base is deleted due to some reason, maybe some type of mutagens, or it can be ultraviolet rays or some certain types of chemicals. If this sequence is, this particular base pair is missing, then you see that AAG will be same because it is occurring after G. So A has vanished, next comes your GUC. So instead of AGU, the next triplet code becomes GUC. Then GUC is, after GUC is CAU. You can see, after GUC is CAU. So let's take for CAU like this. Now the sequence here changes. Suppose it was lysine, so it remains same. Now here it changes. AGU becomes GUC. So instead of serine, valine comes, and CCA, which is a proline, instead of CAU, suppose histidine comes. So that means wherever the deletion has taken place, rest of the frame of this RNA that changes, all the sequence of the amino acid changes. Now this is what we call a Frameshift mutation. That means the whole frame of amino acid becomes wrong because of a base pair which is deleted at a particular point. So this is a type of Frameshift mutation which occurs due to deletion. Same thing can happen in insertion. We take that same sequence, suppose AAG, which is lysine, AGU is serine, AAU is, suppose, aspartic acid. Now here, suppose one particular sequence of amino acid is added here. So the addition is taking place in particular points. Say AAG, AGU is the serine, then AAG. Here AAU is aspartic acid, and one G has come and joined in this particular place. So instead of AAU, it becomes AAG. So you can see here AAG remains same as lysine, AGU remains same as serine. But where it was aspartic acid, AAU, the sequence has changed because of the addition of G at this particular point. So it becomes AAG. So AAG actually codes for again lysine. So aspartic acid changes into lysine, and henceforth the rest of the amino acid also will change because the whole frame changes because of addition of one particular nitrogenous base at this particular point. Now this is another type of frameshift mutation where insertion has occurred. So frameshift mutation can occur due to deletion, it can occur due to insertion. These are the two types of frameshift mutation. Now the second type is substitution. This is another form of mutation where a wrong base is copied. Like it can be a purine which is replaced by a wrong purine. It can be a purine replaced by a wrong pyrimidine. So substitution reaction is of two types, where you can see they are called transition and traversion. Now transition is that if a purine is replaced by a wrong purine, suppose during this replication of the DNA, instead of A is a purine and G is another purine. So instead of A, it is wrongly copied by G. Okay, that means a purine is copied by a wrong purine, or vice versa, G is copied by in form of A, or T, which is a pyrimidine, which is copied by a wrong pyrimidine C, or C copied instead of C, T is copied. So transition is if a purine is copied wrongly by another purine, or a pyrimidine is copied by a wrong pyrimidine, and vice versa, it's called transition. In traversion, this will be just reverse. Traversion is a purine is copied wrongly by a pyrimidine. It's not purine, but purine copied by pyrimidine, wrong pyrimidine, or pyrimidine copied wrong purine, and vice versa. That's called a traversion. Like say for example, A is a purine and thymine is a pyrimidine. So A is copied wrongly by thymine, or T is copied wrongly by adenine. That occurs particularly during the replication of the DNA. Here also, like T copied wrongly by G and A copied wrongly by C. So this type of sequence, I have showed it with arrow. Okay, this is called the traversion. So substitution is, all this leads to wrong sequencing in the DNA, that will lead to wrong sequencing in the RNA, and this leads to genetical disorder. Now this type of substitution occurs during the mistakes that occur during the replication. The third type is chromosomal aberration. We'll study about this in details in the next course, but let us see in the digital board some of these types of mutation in details. So you can see here the types of mutations. There are two types: the point mutation and frameshift mutation. So this is showing a sequence of a DNA. You can see GUC, AG. So all this ATGC, they are sequenced in a form. So let us see what happens. They have given an example. The cat ate the rat. This is just an example. There are three alphabets are there to show that these are the codes of the amino acid. Okay, now see what happens during the point mutation. Okay, this is GUC is replaced, showing that TAG. This is the nucleotide bit. Since this is an example, the example is like this. Now let us see what happens. They are showing all the sequence. Now one base pair, a wrong base pair is coming and joining here, which causes the change in the genetic code. So suppose B comes and joins at this place, replaces C, the whole meaning of the sentence changes. The bat ate the rat. Now this is what we call a point mutation. A particular point, a wrong sequence which is added or replacing some other, and the whole meaning of the sentence. Similarly, in case of mutation also, the whole thing changes after that particular point. So this is an example, actual example. You can see here normal DNA with a normal protein. There is an altered genetic code in the DNA which is a mutated protein. Now this is an example of the sickle cell anemia. Sickle cell anemia, you can see the sequence of the amino acids in the beta chain of the hemoglobin is valine, histidine, leucine, threonine, proline and glutamic acid and glutamic acid. Now see what happened. This is the normal sequence of the beta chain. Now here some wrong insertion is taking place. Instead of TA comes and join. So what happens to the glutamic acid? You see, glutamic acid changes to valine. So if glutamic acid changes to valine, the whole hemoglobin structure becomes an abnormal hemoglobin. So this abnormal hemoglobin gives rise to a type of anemia which is called the sickle cell anemia. And you know, sickle cell anemia is very much prevalent among the some people in the world, particularly in Africa. It has been seen that lot of these people having sickle cell anemia. So here this Frameshift mutation is another example how this addition of the base pair, insertion of the base pair takes place. And you see suppose RK is changing here, T, the cat, arc, tet, her, at, means the whole change, the change after this point where the insertion has taken place, all the other words that change, that is what is called the frame shift, means the frame changes here where after the insertion. Before that it is normal, that means the normal amino acids are produced, after that there is a change in the amino acid. So this is all about the Frameshift mutation, and till then we'll take up the chapters, chapters after which are continuing in the mutation chapter in the next class. Till then you just revise the chapters. If you have any question, you can put up the question. Thank you very much.